Hello, everyone. This is the second part and last part of chapter 2. We are going to start with looking into cell division and death. Why is cell division occurring in our body and why are cells dying? Well, cell division occurs because of the necessity for growth. When you think of yourself, you were at one point a baby, and then you become an infant, you became an adult, and now a full grown adult, right? You've changed tremendously in size, right? You have a number of cells that increased tremendously. The body needs to develop to go from embryo to a fully well developed baby. You have to have cell division occurring. But not only cell division, you also require cell death during the process of development. Cell division and death are also essential for maintaining a healthy body, for healing part of the body that are otherwise sick or injured. The two processes, cell division and cell death, really go hand in hand. Now, we are going to notice that as we consider different parts of our body, not all parts of our body have the same rate of cell division and cell death. Now, let's first of all address cell division. Cell division involves mitosis, which is really the division of the nucleus slash DNA, if you will, the distribution of DNA among the two new daughter cells. Another part that is very important to mention about cell division is cytokinesis. Cytokinesis is the division of the cytoplasm. During cell division, the two new daughter cells have to each acquire every single part of the cytoplasm that is part of the different compartments, playing very specific roles during the activity and the structure of the cell. We're thinking about, for instance, mitochondria. Every single cell has to have mitochondria. We're thinking lysosomes. We're thinking the endoplasmic reticulum. We're thinking the Golgi apparatus, peroxisomes. You name all these organelles. Every single new cell needs these different organelles that are part of the cytoplasm. So the cytoplasm also has to be distributed during cell division. So as you think about cell division, you want to think about one, the distribution of DNA, mitosis, two, the distribution of cytoplasm, cytokinesis. That's for cell division. How about cell death? During the process of development, apoptosis or programmed cell death is a very important process. That process allows the embryo to go from a mass of cell to a perfectly developed human being, right? Mitosis is going to allow an increase in the number of cells in our body, while apoptosis is going to lead to a decrease in the amount of cells in our body. Let me give you an example of where apoptosis is essential. During the development of an embryo to a baby, there is a process by which the webbing between fingers have to dissolve. That webbing is dissolved because of apoptosis or programmed cell death. When the webbing does not dissolve, then we cannot have separate fingers. As you can see in the case of this baby, we call this condition syndactyly, which is that the fingers are stuck together, right? Apoptosis is the process by which this web right here will disappear, and right here will also disappear, so that the fingers can be individual fingers, right? It's the same thing applies to toes. To take only this example just to show you how apoptosis is important during the process of development of humans. Now, let's dive in the cell cycle. The cell cycle can be divided in two major stages. One stage is called the interphase. The interphase is the longest phase in the cell cycle. The second phase is mitosis. Mitosis is actually occurring at the same time as another phase that is cytokinesis, which is the division of the cytoplasm. Now, I told you that interphase is the longest phase in the cell cycle. That phase lasts about 90% of the lifespan of a cell during the cell cycle, while mitosis/cytokinesis lasts about 10% of the time. Now, interphase itself is divided into subphases, while mitosis is also divided into subphases. Now, what are the subphases of interphase? You have three subphases. We have G1 and G2. These two phases are phases during which there is growth and there is a large metabolic activity with the multiplication of organelles. Now, in G1, the cell is preparing for the duplication of DNA. In G2, the cell is preparing for entering mitosis. While in the S phase, S stands for synthesis. Synthesis, but synthesis of what? Synthesis of DNA, right? This is when the genetic information is copied so that you have two perfectly identical copies of the genetic information that are intended to be distributed among the two new daughter cells. Then the M phase is divided into four subphases: prophase, metaphase, anaphase, and telophase. Now, during the mitotic phase, telophase occurs concomitantly with cytokinesis. That's a very important detail to remember. Now, another thing I would like to mention before we go further is that when a brand new cell develops, sometimes that brand new cell is not going to commit to cell division, and it's going to be stuck in G1 stage. Some examples of this kind of cells that are going to be stuck in G1 phase are our nerve cells. Our nerve cells take forever to divide. Now, you've heard that we lose nerve cells as we grow old. Well, in spite of the fact that you heard this, but this is not completely accurate, because our nerve cells still divide. However, the rate of division is so slow that we have the impression that these cells are not going anywhere and that the only thing that happens to them is dying. But yes, you do have nerve cells that are multiplying, right? But they are stuck in a phase that we're going to call G0 phase. And at that G0 phase, in as much as they are in this stage, they're not going to commit to cell division, right? But when they do commit to cell division, then they're going to go on entering S phase and then the G2 phase and the mitotic phase slash cytokinesis. Now, at G0 phase 2, cells are going to commit to either go to cell division or die. And this is when they are going to undergo apoptosis or programmed cell death. So, with that said, let's go back to interphase. Interphase is going to be a phase that is essential for preparing for cell division. The genetic information DNA is going to be replicated or copied. The cytoplasmic organelles are going to multiply. Think of mitochondria, lysosomes, endoplasmic reticulum, the Golgi apparatus, and so on and so forth. It is composed of the G1 phase, the S phase, and the G2 phase. Now, the cell can enter a G0 phase when it is not going to actually commit to any cell division. Now, looking at the interphase and focusing specifically in DNA, we are going to see that in a brand new cell, you're going to have unreplicated chromosome. At the S phase, that chromosome is going to be replicated, right? And as it replicates, we are going to have one copy and another copy of the chromosome. The two are going to be held together at an area that is called centromere. Now, the two chromosomes that are replicated and held together, we don't call them chromosomes. We call them sister chromatids, even though they are still chromosomes, right? But rather than calling them chromosomes, we call them sister chromatids. That's a very important detail to remember, because saying the words sister chromatids is going to tell us that the cell is in a stage where duplication of DNA has occurred. In as much as duplication of DNA has not occurred, we cannot talk about sister chromatids. We can only talk about chromosomes, right? Here we have a micrograph of the sister chromatids held together by a centromere here. Now, let's go in the mitotic phase. The mitotic phase, as I mentioned, is divided into 4 subphases: prophase, metaphase, anaphase, and telophase. Telophase goes alongside with cytokinesis, but we are going to address that later. But what is specific to prophase? Prophase is characterized by the fact that the genetic information, DNA, is going to mingle with proteins called histones, and this mixture is going to pack up so that the chromosomes are going to be condensed so much that we can actually see them under a microscope, right? So one, the chromosomes are condensed and we can stain them so that we can see them under a microscope. We have the mitotic spindle that start developing, and this mitotic spindle are nothing but the cytoskeleton. And the very specific component of the cytoskeleton is going to be the microtubules. Microtubules. These microtubules that are going to grow outward from the centrioles are now going to target very specific area of the genetic information, that very specific area is the centromere. So other thing that is worth mentioning here that is happening during prophase is that the nuclear envelope is going to dissolve, right? That nuclear envelope that dissolves is going to cause the genetic information to be exposed to the remainder of the cytoplasm. From there, the mitotic spindle will keep moving toward the genetic information so that they are going to attach to the centromere. There are particular sets of proteins that are located in the centromere. These proteins are called kinetochores. They are called kinetochores. And these kinetochores are going to be the region where the mitotic spindles are going to attach so that the mitotic spindle are going to pull chromosome toward opposite direction in so much that these chromosomes are going to sit in the middle of the cell. That's why we call this meta. Meta, middle, phase, it's the phase of mitosis during which the chromosomes sit toward the middle of the cell, on the equator line of the cell, right? So the chromosomes align. Then we're going to move to anaphase. Anaphase is the phase during which the mitotic spindle are going to shrink so much that they are going to pull the chromosomes away, the sister chromatids, I should say, away toward opposite direction so that this group of sister chromatids here, group of sister chromatids, at this point, the two sister chromatids are separated, right? So one chunk is going to come this direction toward the centriole, while the other one is going to move toward this direction, toward this other centriole here. All right? So by moving toward opposite direction that way, we are preparing to develop a new cell. So anaphase is the time during which centromeres are going to break, separate, so that the sister chromatids are now going to be separated, right? We're going to have one group here moving toward the centriole and one group here moving toward the centriole. Now we are really ready for the development of two brand new cells. The genetic information has been separated. Now we are going in a stage where we're going to see the development of a cleavage furrow. Right here, you see a cleavage furrow. Now, that cleavage furrow is the sign that we are in telophase. So in telophase, the spindle, the microtubules, if you will, they're going to disassemble. The nuclear envelopes will start reforming. Okay, so you can see the nuclear envelope starting reforming here. Okay, you have a depression here, that is the cleavage furrow. At the same time this is happening, the cytoplasm is also being distributed among the future daughter cells. This is one future daughter cell. This is the second future daughter cell. And each of them, they have to inherit all the different categories of organelles that are present in the cell for its activity. So now we end up with cytokinesis. I already mentioned this. This is the division of the cytoplasm right? The nuclear envelope has been developed, right? Organelles are going to be distributed. The macromolecules are going to be distributed into the two new daughter cells. The microfilaments are going to cause a split of the future two daughter cells into indeed two daughter cells. Now, the cell cycle as it goes in the body has to be tightly controlled. Because if you have uncontrolled cell division, then you go into cancer, right? We have a whole topic on cancer that we are going to address, so let me close that parenthesis. And let me tell you that the cell cycle has to be tightly controlled so that there are checkpoints, right? There are checkpoints. You have a checkpoint during interphase. Here, the first checkpoint in G1 phase, that checkpoint is going to be essential to control if the brand new cell is okay. Is okay with the DNA being clean, fine, and the number of organelles being okay in terms of the category of number of organelles. For instance, if a cell by chance is missing mitochondria, how is that cell going to produce energy for the metabolic processes? That cell will not be able to reproduce. Therefore, that cell will have to undergo apoptosis, right? So you understand that that cell has to be checked to see if it's okay or not. So we have a checkpoint there. We're going to have another checkpoint at the end of the synthesis of DNA to find out if that DNA was properly synthesized. Then we have a checkpoint that is going to check in, now that we know that the DNA has been properly synthesized, there is no major mutation, or if there were mutations, these mutations were corrected. Now that cell is going to commit to enter cell division, right? Mitosis. So it goes in mitosis. And when it goes in mitosis, it undergoes prophase, metaphase, anaphase. Now, before actually the cell undergoes anaphase, there is a control that happens there. The cell is going to verify that all kinetochores have a mitotic spindle attached to them. If all kinetochores do not have mitotic spindle attached to them, there is going to be a stop to the cell cycle. So there is a control for the cell to go from metaphase to anaphase. Always. Only cells that have all the mitotic spindles the way they should be are going to move on to anaphase and telophase and cytokinesis. Any cell that is not properly structured like that is not going to be able to enter the phase. Okay? So there are checkpoints. That's one thing you want to know, right? These checkpoints are checking for the integrity of the cell, integrity of DNA, and also is the DNA going to be able to be distributed properly with the cell, the daughter cells, being prepared to receive the exact same amount of genetic information, right? So now we have inhibition of the cell cycle so that DNA can be repaired. If that fails to happen, the cell will undergo apoptosis, right? You have you have a number of factors that are going to check at the end of during the G2 phase and at the end of the G2 phase to see if everything is ready for the cell to undergo division of the nucleus. If not, the cell is going to undergo apoptosis or it's going to stop cell division altogether, right? And then you are going to have the attachment of spindles, okay? So you have a number of verifications that are going to happen here so that the cell is going to be controlled to go in anaphase or not, or else there is no cell division moving forward. We have a stop to the cell cycle. All right. Now, the cell cycle is limited in all cells in our body. In other words, what I'm trying to say is that a given cell can multiply so many times. And the question we may ask is, why are our cells limited as to how many times they can multiply through their lives? Well, the reason, and we find out that it's because the chromosomes have at the end some sequences that are repeated. The sequences that are repeated are called telomeres. We notice that after one cycle of cell division, the telomeres shrink. The telomeres are made up of repeated sequences that are TTAGCC. TTAGGG. That's the sequence, TTAGGG. This TTAGGGs, it's multiple repeats. And every single cycle, our cells are going to lose those repeats so that they shrink, right? Lose 50 to 20 to 200 after each cell division. So much that after 50 divisions, well, the chromosomes will be so short that beyond that, the chromosomes will start losing vital information for integrity of these chromosomes. Now, let me tell you that the telomeres actually do not encode for nothing specific in our genome, right? However, these telomeres are there to protect the genetic information that is within the chromosome. So how you want to picture this, it's basically that you have your chromosome, like here, containing all the nice genetic information. The exomes, right, are going to be in all this area. And then you have a set of sequence of DNA here that doesn't encode for anything, but these are telomeres. And these telomeres are going to be lost progressively as cells divide, so that there is a certain point in time when, as the nucleotides are lost, now we are starting to attack the genetic information that is essential in here. Okay? So the cells are going to be able to grow, but they are going to obey a number of signals. Whenever, for instance, in the lab, we grow cells in plates, these cells can only grow so much. There is what we call contact inhibition that is going to happen, right? So if you have a petri dish here, and you have some cells on that petri dish. These cells can only multiply until they reach the edges. And we notice that these cells will not start stacking up like this. They won't do that. Okay? They will go up to the very edges of the plate, and that's it, right? We call this contact inhibition. And that contact inhibition is what controls the growth of the cell, right? Sperm cells, egg cells, the bone marrow, and cancer cells can reproduce continuously. They can reproduce continuously because they are expressing an enzyme that is called telomerase. This telomerase is responsible for preventing the shortening of telomeres. Cancer cells express telomerase. That's why these cells are immortal. Sperm cells produce telomerase. That's why a male can produce human sperms all the time, right? And egg cells also have this. And notice that sperm cells have telomerase, egg cells have telomerase. This comes without real surprise when we know that sperm cells and egg cells come together to form a zygote. That zygote is the one that is going to undergo multiple cell division to form an individual until you are completely developed, you are born, and you grow up to the age you are. If you consider your skin cells, for instance, your skin cells are going to divide over and over and over more than 50 times. Okay? More than 50 times, more than 100 times. And this is possible because at one point in time, right, your sperm cells and your egg cells, they have this telomerase, and this telomerase will be active up to some point in our life. And at some point, these telomerases are not expressed anymore. And when this telomerase stop being expressed, this is now when your cells will see their telomeres start shrinking. Now, hormones play a very important role in cell division, so do growth factors. Hormones are going to be made by glands. They are released in the blood and transported to different places in our body. Hormones are characterized by the fact that they have very specific effects and they attach to very specific receptors on the surface of the cell. On the other hand, growth factors are these chemicals that act locally. They are not like they are not like hormones that actually travels through the blood, but growth factors are going to be produced locally. For instance, epidermal growth factor, EGF, is going to be released when we need to replace skin cells, right? For instance, when you have a cut, right, that cut is going to be repaired because of EGF, epidermal growth factors, right? Now, in the control of the cell cycle, there are two major proteins that play a key role in the cell cycle. One is the cyclins. Cyclins are proteins that are produced during the cell cycle. That's one protein. Another set of protein is the cyclin-dependent kinases. The cyclin-dependent kinases are enzymes, right? They are enzymes. They are called kinases because they allow phosphorylation. They're going to allow the attachment of a phosphate group to another molecule. So this cyclin-dependent kinases, CDKs, cannot work unless the cyclins are present. That's why we call them cyclin dependent, right? If cyclins are not there, forget about their activity, right? So this complex cyclin dependent kinases is going to be essential in the control of cell division. Okay, they're going to interact with each other and control cell division that way. The cyclin and cyclin dependent kinases are going to activate cell division by causing genes essential for cell division to be expressed so that mitosis can occur. So I want you to remember the role of the cyclins and the cyclin dependent kinases in cell division. I'm not going to go into the intricacies of that. I just want you to know that you have a complex of two proteins, cyclins and cyclin dependent kinases, that when working together are going to stimulate cell division. Now, how about apoptosis and necrosis? Apoptosis is programmed cell death. Necrosis is cell death, but it's not a programmed cell death. Necrosis, it's basically when we lose cells because the cells are damaged, right? They can be damaged because of a toxin, because of irradiation, because of burn, because of many reasons. But definitely we see necrosis when there is issue with our health, while apoptosis happens in a normal, healthy body, right? So, typically, a normal cell, right, will be tagged to be destroyed because there is something going on with that cell. Say, for instance, you were exposed to a heavy amount of UV light and the DNA in your skin cell got damaged, right? Well, these cells will die by necrosis. That's one thing. But on the other hand, you have that when you consider the web between fingers, that web has to disappear. These cells are going to receive a signal. The cells in the web are going to receive a signal that commands these cells to undergo apoptosis. So typically, the death receptors on cells that are doomed to be destroyed going to be activated and cause the activation of a particular group of proteins that are called caspases. These caspases, which are enzymes, are going to destroy a number of proteins in the cell. That destruction of this protein is going to cause these cells to lose shape and to become undulated, as you can see here. So the cytoskeleton will start getting disorganized, the nucleus will be disorganized, and you will see some blebs forming. These blebs ultimately are going to lead to cell fragmentation, and the cells that are fragmented are going to be engulfed by some of your white blood cells, specifically phagocytic cells. And these phagocytic cells are of two kinds. We're going to have macrophages And we are going to have the neutrophils. So these two phagocytic cells are going to engulf all the cell fragments and cause a destruction of the cell that way. However, that is apoptosis. However, in the case of sunburn, the cells are dying because the cells were damaged, right? And the damaged were so much that there was no possibility of repair. These cells are going to die. All right? So the caspases are going to specifically target enzymes that are involved in DNA replication, okay, and DNA repair. They're going to target the cytoskeleton. Cells are going to lose the ability to attach to each other, and they are going to attract phagocytic cells, including macrophages and neutrophils, for their destruction. Now, for new cells to develop, and when you think about cell development, these cells are going to come from stem cells. Stem cells are blank cells. We saw that in the first chapter, right? These blank cells have this ability that they can self-renew. That's a very important property I want you to remember. That ability of self-renewal is going to allow the body to always have a batch of stem cells to go from. Otherwise, some cells are going to undergo partial differentiation. So they're going to be partially specialized, so that we call the cells progenitor cells. Progenitor cells are the stem cells that are already committed to some level of differentiation, but they are still blank cells. Nevertheless, these blank cells are designed to become very specific kind of cells in our body, right? For instance, there are progenitor cells that can only become blood cells. They cannot become skin cells, they cannot become muscle cells or nerve cells, right? Okay, now you have these progenitor cells. After they receive a specific signal, they're going to turn into blood cells. They can turn into white blood cells or red blood cells. Everything depends on the kind of signal they received, right? If they receive the factor what is it called? CSF colony stimulating factors, colony stimulating factor, okay, they are going to cause the CSF are going to cause this progenitor cell to turn into white blood cells. There we go. By the same token, if the signal is EPO or erythropoietin, this progenitor cell will not become white blood cell, but it will become red blood cells. Red blood cells. The stem cells are going to be of different kind, right? You have the stem cells that are absolutely blank, and you have stem cells that are progenitor cells, right? So we have those ones that are blank, And these ones are partially blank, okay, because they are already committed to become only a very limited number of cells. So that when we look at the capability of these cells to become differentiated cells, we say that some cells, some stem cells are totipotent. Stem cells that are totipotent are true stem cells. These cells can develop any kind of other cell in your body, whether you're talking about skin cell, you're talking about liver cell, brain cell, muscle cell, kidney cell, bone cell, you name it, right? Then you have some blank cells that are pluripotent. These blank cells that are pluripotent only have the ability to become a limited number of cells. For instance, becoming, say, red blood cell or white blood cell or platelets, right? These are pluripotent. And then you have multipotent. Multipotent are going to allow the cells to develop into different kind of cells, but this is only happening during the process of development from the embryo, right? So many organs, if not all the organs in the human cells, are going to have stem cells or progenitor cells. Now understand that these stem cells or progenitor cells are already predetermined to become a particular kind of cell, right? Very important to remember that. So when we go back from the beginning, we have a sperm cell and an egg cell. They come together to form a zygote, right? That zygote, which is a fertilized egg, is going to give rise to stem cells here. These stem cells here have the self-renewal ability, right? They have the self-renewal ability. These cells are literally immortal. They can divide over and over and over and over. They expressing telomerase, right? Then these stem cells are going to partially differentiate into progenitor cells. And notice that a progenitor cell here is not the same thing as a progenitor cell here. And again, a progenitor cell here is not the same thing as a progenitor cell here and here. Why? Because we notice that while this progenitor cell here, right here, can differentiate into many different kind of cells, but we have that this gives rise to this progenitor cell. This is a multiple step process. And then this progenitor cell is itself going to give rise to progenitor cells that can either give red blood cells or give white blood cells. This progenitor cell right here can give red blood cells or white blood cells and the platelets. So the progenitor cells right here can only give blood cells. Now, on the other hand though, when we look at the progenitor cells right here, we see that this progenitor cell can differentiate into other types of progenitor cells, that some are going to give rise to fibroblasts. These are cells of the loose connective tissue. and cells that are going to become bone cells. So we have future bone cells right here. Now, understand that going from stem cells to progenitor cell, it's a process, multiple stages, right? Okay, and we see that some progenitor cells are closely related compared to other progenitor cells. Now, for instance, the red blood cells and platelet cells are more related to the connective cells than they would be to nerve cells or skin cells, right? Everything depends on where the separation started happening. And that separation, that specialization, it's because of the type of signal cells are receiving during the development or during growth, right? So that's basically is the pathway of cell specialization. And the question we may ask is, what is the source or what are the sources of stem cells? There are really three major sources of stem cells. We have the embryonic stem cells. Embryonic stem cells are the cells that can become about any other kind of cell. These are totipotent cells, right? So they can be obtained from an embryo, right? Or somatic cells in which you have a nuclear transfer into an egg. But that has to come from an embryo, basically. Then you have cells that are stem cells because they are induced pluripotent stem cells. And this induced pluripotent stem cell, this has to happen in a lab, right? There, genes or other chemicals are going to be used to reprogram the cell. Okay? To reprogram the cell, typically it would be, for instance, your skin cell or your muscle cell. It has to be a somatic cell, but definitely not from an embryo. Okay? It has to be a somatic cell from a grown individual. And then you have adult stem cells. These adult stem cells are somatic stem cells that have a special function that can be isolated and be used to basically modify an egg, right? So these are different sources. So the embryonic stem cells, induced pluripotent stem cells that are modified in a lab, and adult stem cells that can be obtained, for instance, from the bone marrow. Okay, that's an example. Then what is the purpose of stem cell? Now we hear a lot about stem cells. There are really many purposes to stem cells. Ultimately, it boils down to medicine, solving our problems. So we're going to have a donor stem cell that can donate stem cells to repair an organ in a person who is sick. For instance, someone with cystic fibrosis or someone with sickle cell disease, just to name these two examples. Then you have that a fully grown individual can actually donate its own cells to actually solve some of the problem. For instance, to do skin repair when someone is burned, right? Rather than taking skin cells from another person where there is a chance to see tissue rejection, we are isolating cells from a patient and using and culturing the cells in lab for the repair of the body. And then we have reprogrammed cells. And again, this is in a lab where some cells can be isolated, right? Use some chemicals to reprogram the cells to cause them to become blank, and then stimulate them with very specific growth factors, causing them to become red blood cells, muscle cells, or nerve cells. Now, these processes are all possible these days. However, the technicality of making this happening is what limit the availability of these resources in medicine. So again, the purpose of stem cell is going to be for discovering new ways of diagnosis or treatment or developing new drugs, to study the early sign of disease, create tissues for organ transplant, okay, stimulate stem cells in the body to reprogram them and form proteins. We have more and more also some people considering stem cells to stay young. Okay, now obviously when we start digging into issues like staying young, then we are entering into questions of bioethics, right? Should the stem banks be regulated? If so, how? The companies that are involved in stem cell research, what kind of information can they post on on their website, on the internet, and what kind of information should they not post, right? So, what can we do with the stem cells, and where do we obtain from stem cells? Now, you heard about the embryonic stem cells, where the controversy is, okay, well, should we devote some embryos to research? If so, how and who should do that? You know, the discussion is ongoing and still very controversial. Now, finally but not least, let's address the microbiome. Now, in our body and on our body, believe it or not, we have more microorganisms than our own cells, right? Now, mind that about 60% of the cell in our human body are microorganisms, right? That's a lot, 60%, right? So all these organisms that live in us and on us constitute the human microbiome. It's becoming more and more important these days to understand the microbiome, because we notice that it plays a major role in our health and the quality of our life, right? Different body parts are going to house different communities of microorganisms. That's something we've learned. For instance, your hands are going to have different sets of microorganisms compared to your mouth, compared to your ears, and other parts of your body. Okay? And also, the microbiome is going to change based on our personal experience, based on where we live, based on what we eat, right? Based on what we do, right? Now, if you consider the lifespan of an individual, An unborn human has zero bacterium, zero virus. They are sterile, literally. Humans start getting exposed to the microbial world when they are born at birth, right? First of all, through the birth canal, the birth channel, okay? The vagina. The vagina has a certain amount of microorganisms that are going to stick on the baby when the baby is going through, okay? And most likely too in the baby because the baby can absorb some fluid, right, as the baby is coming through, right? So that we have a certain number of microorganisms. Now, from birth to old age, the composition of the microbiome is going to change. First and foremost, the baby will be breastfed, right? When we look at the distribution or the proportion of the different microorganisms, we see that, for instance, in a baby that is breastfed, the actinobacteria and the bacteroidetes and also the proteobacteria are going to be abundant in there, while other bacteria are going to be present in very limited amount. But take a look at this. When a baby starts eating solid food, we see that the amount of other bacteria is going to tremendously increase. The proportion of actinobacteria is going to decrease, while the bacteroidetes is going to increase. So you have a change there, right? When the baby grows to become a toddler, the type of microbiome is also going to change. Look at a toddler after an antibiotic treatment. We see that we're going to have the bacteroidetes that are going to be so many, while other microorganisms are going to be destroyed. Okay, so an antibiotic treatment affects the microbiome. How about malnutrition? Yes, malnutrition is going to affect your microbiome compared to when you are healthy, right? An adult being healthy and being obese is going to affect your microbiome. And when we grow old, based on how old we are, our microbiome is also going to be different. So the human microbiome has revealed a number of things. For instance, certain skin bacteria cause acne, but others are going to keep the skin clear. Circumcision has shown to protect humans against some infections, including HIV. Lowered blood sugar following weight loss has been observed, and this is partly due to a change in the gut microbiome during the surgery. Whenever microbiome is altered, this has hastened starvation in malnourished children. Antibiotics, they affect the microbiome in the gut, not just in the gut, but also in female, in the vagina, for instance. Okay, that's why when a female undergo a heavy antimicrobial treatment, right, there is a particular organism in the vagina that is called Candida albicans. Candida albicans start growing out of proportion, and that's why a lady is going to feel itchiness in her private part, because lactobacillus which is an organism, a bacterium that causes the vagina environment to be acidic, is not there to create acidity. Therefore, a Candida albicans, which is a yeast, starts growing out of proportion. To mention only these examples. Another thing that is worth mentioning is that the microbiome in babies that are born through the vagina is not the same microbiome as babies who are born through C-section. Okay, very different. And people living in isolation do not have the same microbiome as people who are really circulating everywhere. So this actually concludes this chapter. Thank you for your attention.